Thanks, Reza, for that very nice preparatory talk, I would say. I can right away skip at least 3 of my slides, I guess, because the aerial tomography workflow has been so nicely introduced already. So I think then it may make sense to just focus a little bit more on what the multi-sem actually can do. And maybe one other thing before I forget, if you happen to come to Oberkochen to look at the ZEISS BSD detector, please be welcome, be our guest, and also visit the ZEISS MultiSEM, which is in the same demo facility. You see it in my background. I'm not actually there, so it's just the virtual background because we have some maintenance going on in the demo lab with the air conditioning. Therefore, it's not going to be a live presentation, which I would have loved to do. But I just pre-recorded a couple of video takes from the workflow, so I hope that will do the job as well. So let me start with, well, I think this is maybe well known to most of you. So brain research is a megatrend since I would say at least a decade, maybe even longer. One special field within the neurosciences, namely connectomics, is one field that is very important to us because it's the first field that got interested in an ultra-fast scanning electron microscope. So if you would look at connectomics in a broader sense, you can talk about connectomics in various scales. The macro scale, which would be in millimeter spatial resolution. Usually the technologies used here are fMRI or anatomical MRI imaging or diffusion tensor imaging. The nice thing, of course, with that scale is you can use living subjects. You can work in vivo, and that's, of course, very advantageous. On the other hand, sometimes you want to look a little bit closer, and then you're entering the mesoscale with hundreds of micrometers resolution, where people use wide field light microscopy or X-ray microscopy, also optical imaging, and this is then getting more and more invasive. But if we really want to know how neurons are connected to each other, how individual neurons are building networks, then we really need to look a little bit closer even and go down to the nanometer scale. And there are light optical methods here as well, of course, confocal microscopy or super resolution microscopy. However, in these technologies, you only see what you're labeling. And sometimes it can be really useful to have the full picture and to be able to densely reconstruct certain volumes. And then why would it be necessary to speed this up, to have this on a faster scale? That of course makes a lot of sense if one looks into the orders of magnitude that the components of the neuronal systems are actually spanning. So I mean, there are individual neurons that are about 1 meter long in the human, namely going from the brain down the spine. Of course, in most animals, it's maybe less. It's maybe just 1 centimeter, or half of a centimeter going from one hemisphere to the other. But bringing together these dimensions of hundreds of micrometers or maybe even millimeters, and the requirement to spatially resolve that with a couple of nanometers in pixel size, and to be able to also identify ultrastructural details like synaptic vesicles or intracellular organelles. And then, of course, it makes a lot of sense to think about speeding up electron microscopy. And if we look now at the portfolio that ZEISS is offering, all these orders of magnitude I just mentioned, of course, can be tackled with different methodologies. However, you always need to switch the modality then if you enter another scale. And the multi-sem is maybe kind of a bridge technology between the less well-resolved but large area imaging systems, light microscopy or X-ray, and the very high resolution electron microscopes, the standard field emission electron microscopes, or even a helium ion microscope. And now, if we look at the topics one can address with such a system, then of course neurobiology is maybe at the moment the most important one for us, because most of our customers are working in this field. However, there are different fields of research that can make use of such a system as well. And as the Multi-sem is a scanning electron microscope, it's not operating in transmission mode, like, for example, the FASTIM ophthalmic. Other materials can be imaged just as well, being it microchips or solar cells, battery materials, electronics components, or also block phase materials in biology, of course, like, for example, bone samples, or also block phase samples if they are conductive enough. And the system itself comes in two flavors, one with 61 and one with 91 beams, which is maybe a minor difference. It's just one is a little bit faster than the other one. And fast in that sense means it's about a terapixel per hour that the system can generate data. The spatial resolution is about 3.5 nanometer, which is well suited for most neurobiological questions. And of course, it only makes sense if it's mainly automated. So you don't want to sit at a high-speed tool and fiddle around with some knobs. So it needs to run more or less autonomously over a certain amount of time. So how does this now work? This is a short video clip giving an idea of the operation principle. The Multi-sem starts with a single field emitter, just as any single beam system as well. And we send this beam, it's broad and it's sent through a multi-hole aperture, basically like a shower head, my former boss used to say. And then this array of beams is scanned over the sample surface globally. And the secondary electrons that are generated on the surface are collected with the objective lens and projected into a second column. So we have an illuminating and a detection column. And the signal electrons are then projected and focused onto a scintillator, and the actual detectors are photodiodes. So while the beams are scanning over the surface, this bright spots onto the scintillator fluctuate in intensity, and this in the end gives you the signal. And if we compare a single beam system with a multi-sem, then this basically is the whole trick. We just image a much larger field of view at the same time. And of course, not only one of these multi-beam fields of view can get imaged, the stage can be moved as in any other microscope, and so larger areas can be acquired as well. This is an example of a square millimeter, and the imaging time for such an area would be like less than 5 minutes. You can access this video either with the link that's written here or with the QR code. So it's public, yeah, if you like to look at it again, or if maybe it didn't show up well in the presentation here. And in the static view, this would now mean at the left side, we see a picture of that scintillator where the secondary electrons are hitting and producing a photon signal. And each of these bright spots is generating one of the subfields of the multi-beam field of view. So the center beam generates this center fraction of the field of view, and the other encircled beam generates the image on the lower right, and all the other beams generate the other images. And then these individual image tiles are merged to build this hexagonally shaped multi-beam field of view. which is about a little bit more than 100 microns across in the 61 beam version, and it's about 165 micrometers from left to right in the 91 beam version. And this is the area that is imaged with one scan pass, so in a couple of seconds. And as an example here now is a full section of a cortical section of mouse brain. Just for your orientation, on the upper right, the laser pointer here. So here on the upper right, this would be the cortical surface. This is the white matter. We have the ventricle here. So here is the midline, and this is the gray matter. And this full section is about 3 square millimeters and is imaged in about 20 minutes, a little bit less. It's about 300 gigabytes in size. And this is basically what you can expect. Now, if we do the calculation for a little bit larger volume, because it's about volume electron microscopy here, it's not about single sections. So if we think about a cubic millimeter of tissue, which is sectioned at 50 nanometer section thickness, then we end up with 20,000 square millimeters that needs to be imaged. And in a single beam system, this needs roughly 2.5 hours. Really depends a little bit on the individual settings, but I think most of you would agree that this is a reasonable time for a single beam electron microscope. However, if we now calculate the full cubic millimeter, this would be about 6 years of imaging time. And this is just beyond doing it. So most people won't start projects with this size. And with parallel multi-beam image acquisition, this can be reduced to about 3 to 4 months. And now we're talking, I would say. And now, again, thanks to Reza, I don't need to go into very much detail here. The perfect match for the multi-sem definitely was the advent of automated sample preparation methods. I totally agree that very skilled people can produce large section series manually. However, it's extremely helpful if you have a little bit more automation here. So the atom tome would be one way to do that, the tape collecting. But I also totally agree that the tape material is an issue. It's not that easy as it sounds. However, there are other technologies being developed, like the MAXI, the magnetic collection, which is also improved by other labs. So here we have now a set of tools that facilitates the production of serial sections quite tremendously. And if we now, there should be a video. Let me quickly start that. Oh, okay. All right. So this is just distracting a little bit in the background. The capabilities and the limitations of the multi-sem is a thing one would also need to talk about, because of course this unique speed comes with some limitations. So the first thing, how do we actually achieve that, or what is the idea about parallelization of the electron beams? Because there would be other ways of speeding up an electron microscope. One could, for example, just scan faster. The electronics are there. One can just scan with not like maybe 20 megahertz, but make it 800 or something. However, you still need to get enough signal. So if you speed up the scanning, you would also need to increase the beam current to get the same signal out of the sample. And if you increase the beam current, then you will lose spatial resolution, because these charged particles will repel each other. So by using parallel electron beams, one can use more current without losing that spatial resolution, because the individual beamlets don't have that current. And then the next limitation one could run into is the detection bandwidth. They usually also have kind of a time resolution. which they need to recover. So if one would just speed up the scanning, at some point in time, the detector will not keep pace with that. And again, by parallelizing that, less detection bandwidth is actually required. And by the unique principle of the multi-sem using an extremely strong stage bias, we basically detect 100% of the secondary electrons. So we have an extremely high signal yield, which allows us to decrease the scan time, the dwell time, to maybe 100 or 200 nanoseconds for a typical life science sample. And this already gives an idea of some requirements to the sample, which can be seen as limitations. The samples need to be flat. Reza already talked about that. For the multi-sem, this is even more important because of the secondary electron contrast. So every wrinkle, every fold shows up much stronger. And this is also the reason for the samples needing to be conductive, because charging effects will impair the image quality quite a lot, which all has to do with the secondary electron detection only. And now it doesn't freeze. Computer shows something all the time. So I will skip this slide because we will just have a look into how it actually works in a live video. So just to give you an idea, the software we are using with the multi-sem is ZEN. This is the same software that ZEISS uses for light microscopes. So if you are familiar with the ZEISS light microscopes, most probably will also be able to use the multi-sem more or less right away. It's a little bit like Office. So if you know how to use Word, you will find in PowerPoint, you will find the same controls, the same positions, and they will do the same thing. And this is a little bit the same here with this software suite. So this is how it looks like in our demo lab. As I said, you see it in my background as well. So we have a multi-sem 506 sitting there where we do sample work. And of course, we also welcome visitors for a live demonstration on site. And also part of the workflow in our case is a light microscope. So an X-ray imager vario, to be precise, it's shown here. And we're taking an overview image of the sample before we put it into the multi-sem, because that's one special thing on the multi-sem, we cannot zoom out. So for orientation purposes, it's really helpful, or it's almost required for this workflow, to have a navigation help, namely a light microscopic overview image. In principle, it could also be a picture of a mobile phone or a normal photograph, but then the spatial resolution may not be good enough to find the sections and define the regions of interest, which we can imagine if we have a look at the sample holder here. So give you an idea of how large that is, this square piece of silicon wafer we have on the sample holder here is 10 by 10 centimeters. And in 3 corners of the sample holder, we have the so-called L markers. So these are little plates that show a large and a small L-shaped engravement. And these markers are registered in the light microscope and then afterwards in the electron microscope as well. And by that, the overview image of the sample can be mapped, can be registered to the electron microscope stage, and therefore the navigation is really precise in the end. So we can use this an overview image here to target the individual sections and the region of interest on these sections. So when we start working on such a sample, we put it on the light microscope first. And on the light microscope stage, we have a receptacle that takes the sample holder nicely in the same position all the time. So even without actually registering these fiducials, you still will get some reasonable targeting in the end. So it's not even that necessary, but if you're doing a real experiment and you're acquiring real data, then of course it's better the more precise it is in the end. And then this is speed up quite a lot. On the left side, one can see taking this overview image of the full sample holder takes about 40 minutes. So this is now fast forward, just to show you how the image will look like in the end. And what we also see, and Reza mentioned that as well, the sections do not always lie straight in line on that tape. I must admit, this is a really nice sample, so the sectioning went really well, and they are very straight. The sample comes from Harvard, and I mean, they kind of invented this device, so maybe not. And wonders not that much, but still there are some sections that are not lying straight. However, on the multi-sem, this actually is not that much of an issue because the algorithm that detects the sections will find them anyway. So we'll see that in a minute. First, the sample needs to be put into the multi-sem. This is the standard airlock, as the same as it is used on single beam systems as well, 200 millimeter airlock. So in principle, one could also introduce the sample through the chamber door, but then pumping down will just take longer. And here it's just a couple of minutes from introducing the sample until you can start imaging. So my colleague Tomasz, he's our application specialist. He's introducing the sample to the chamber now, pulling back the rod, and then pumping down the airlock again to close the valve between airlock and chamber. And then the first thing one is doing, well, while looking at the operating system of the multi-sem. So this is now a screenshot from Zen for multi-sem. On the upper right side, we see an image from the chamber cam, which is sitting in the back. So here we see the sample, and on top we see the objective lens. If you know how the objective lens of a single beam system looks like, this here is way larger and very flat. And this again has to do with this strong sample bias. So not to produce arcing between the stage and the objective lens. And on the hardware status overview, one can see right away in which state the system is. So if everything shows up green, here we see the different vacuum compartments of the system, the chamber, the illuminating column, the detection column, the gun area, and all the other components of the system would also have a drop-down menu where you can look up each of the components to see in which state they are. And if everything is green, one can start working. And we are usually using for this kind of experiment the so-called experiment setup wizard, which guides you through the experiment setup step by step. And one of the first things to do in the serial section workflow is to define the ribbons, which is done here at the moment. And these ribbons define the area in which the section detection algorithm is searching for sections in the end. So it makes things a little bit quicker. So we are now delineating one template section. And by defining a certain range of angles the algorithm should take into account, one can account for sections not lying straight on the tape, but being rotated a little bit. And then we apply. Again, I must admit, this is speed up a little bit. The algorithm will not take extremely long, but it takes, for a full wafer like that, it will take a couple of minutes. And of course, I wouldn't want to torture you with that here in this presentation now. So as you see now, all the sections have been detected very nicely. If this doesn't work as nice as it did here, if it finds sections that are not there, or if it misses some sections, then of course this can be adjusted manually very easily, just as with the Atlas software as well. And then this last step was, if you don't want to image the full section, but only a certain region of interest, then this region of interest is delineated and applied to all the other sections. It could also be 2 or 3 regions of interest. That actually doesn't matter. You can set it up all at once. And then one jumps to the next step of the experiment wizard. For the time being, all this was done on this light optical overview image that has been imported from the light microscope. But to transform this now, this overlay basically, into stage coordinates, we need to import that to the stage coordinate system. And we saw that in the background now, that all these regions of interest from the light microscopic overview image now has been exported to the tiles setup, which is the stage coordinate system of the Multi-sem. And this overview image is also added to that view, just to be able to check if the regions of interest are actually showing up in the positions where they should. And in the next step, okay, you saw that I skipped now a lot of steps because they're just not very interesting to watch. It's basically adding a data path, describing the sample, setting a couple of things within the software, if overview images shall be generated, if the samples, the individual images shall be stitched or not, and with which protocol, and so on. However, going through all these steps will take a couple of minutes if you're a little bit experienced. And now we're coming to a more interesting one. One question that we had earlier during Reza's talk was, how is the focusing done if the sample is not flat, or if the sections are not on the same Z-plane within the microscope, within the chamber? And totally correct. It's very hard to ensure that the samples are actually on the same height. And what we're doing in the Multi-sem is setting up the experiment with an autofocus workflow. And one method would be, I think I just jump back for a second, because I think we skipped that now. So what we do in this workflow is we're setting up an autofocus map. So we are defining focus support points. And here they are sitting outside of the region of interest, so that we don't have contamination from the focusing. So we set up focus support points outside the region of interest. We delete the one here in the middle. You can set this up manually just as you like and as it is required. You need at least 3 of these support points, of course, otherwise it will not be able to calculate a plane. But if it's a larger area, or if it's a sample that has more topography, then of course it can be much more than just this 4 support points we use here now. So this is really up to the actual sample that is used. And then the position of these support points is copied to all the other regions of interest as well. And in this video, we now see how it works when everything is finished. So the experiment is set up. The system asks if everything is fine and can start imaging, and then actually it starts imaging. So it starts by targeting the focus support points one after the other. And there it's now measuring the focus and the astigmatism at this position, basically by just going through a range of focus steps and astigmatism values, and then retrieving the optimal settings for that position. And if it turns green, then it was successful. Going to the next support point, doing the very same thing. It's doing this for all of these 4 support points. Then it's calculating a plane. There are various algorithms one can choose which type of plane shall be calculated. And then it starts imaging. So if you zoom in a little bit further, we see these hexagonally shaped areas that tessellate the region of interest. And each hexagon that turns green is a hexagon that has been imaged and successfully saved. On the right side, we see a preview of the data that is generated. And somehow my computer wants to tell me something. This is a compressed view, so this is not the actual raw data because it would be too large. But it gives you enough information to make sure that it's working. It's working and it's imaging the sample, and there none of the channels are just black or something. So it's working nicely. Again, the video is sped up a little bit now. In the beginning, it was real time. It takes about 1 or 2 seconds per hexagon, and then about a second to move to the next position. And if we look now at the folder where the data is generated, we generate this in a hierarchical way. So we have a folder for the region, and then we have a folder for each of these hexagons. And in each of these folders for a hexagon, we have the individual image tiles. Which will open now in a minute. Each folder is about 750 megabytes. So each image has about 8 megabytes. File format usually is bitmap, can also be TIFF or something else. And as you see, besides the actual raw image, we also generate a thumbnail, a much compressed version of that image just on the fly, because we use it for this overview image. And 2 text files are generated. Now that was a little bit too quick. Go back a second. There's 2 text files generated. One that shows all the imaging parameters, the settings of the microscope, the operator, the sample name, things like that, the size of an individual image. And the second text file that lists the X and Y coordinate for each of these individual image tiles. And this format can be directly imported to Fiji, so to the TrackEM2 plugin that Risa also mentioned. So the file format and the file structure, the data structure, is generated in a way that makes it as easy as possible to communicate with open source software packages that most people in the field are using. So this is not a proprietary image file format. As I said, it can be bitmap, it can be TIFF. You can set this during the setup of the experiment. So, and this is an example of how the data looks like. Now the zooming in and out is a little bit quick here. So maybe here we can acknowledge it a little bit better. So this is raw data. It's not inverted. Usually people invert the contrast to resemble it a TEM image. The raw data, of course, is not yet inverted, but for presentation purposes, people used to do that still. Okay, and that's how it works. It's actually pretty boring to watch in the end, because if everything is set up and it's running, it's just watching hexagons turning green. And unfortunately, it's not as fast as we see it here. This, of course, is speed up a little bit again. But I hope that gives an idea. So, and when everything is finished, so now we see here, well, 14 of these sections are already imaged. In addition to the raw data, one overview image is generated as a single file. So this is generated out of this thumbnail images, out of the compressed images. However, if we zoom in after a certain point, it will switch to the raw data. So this is a little bit like the Google Maps approach. You have the compressed view when you're zoomed out, and when you zoom in, you at some point it will take the raw data from the file storage, and yeah, one can have a look at the actual data. What we here see also, this is maybe not the, well, it is a quite nice sample. However, we see a couple of wrinkles, especially in the myelin sheath here. And as I said, in secondary electron imaging, this shows up quite a lot. On the other hand, secondary electron imaging is way faster than backscatter imaging. So once you've optimized your sample preparation protocol to be able to use secondary electron imaging, I think it's advantageous in the end, because you can image much faster. Okay. And yeah, I mean, this is maybe well known to all of you. What you do in the end with this data, of course, is segmentation and reconstructing it in 3D. This software is not part of the Multi-sem. So at the moment, it's still in the hand of our customers what they're doing with the data. And many of them are using Fiji, at least on a low level. When it then goes to larger, well, this will not play. Well, I think you most probably have seen these two renderings anyhow already numerous times. So let's go. Okay, well, when it then comes to larger data sets, not only these small cubes that was just shown in the previous slide, then of course Fiji will not do the job anymore. Jeff Lichtman, our first customer at Harvard University, is collaborating with Google in the meantime. They have a team of neuroscientists who are working on reconstructing and segmenting this type of data sets. There's one preprint available with a human data set, 5,000 sections of 2 by 3 millimeter, a total volume of about a cubic millimeter and 1.4 petabyte of data. There's behind the QR code, you will find a Google block entry, which is really nice to read, and which also includes links to the actual data set on Neuroglancer, which one can play around a little bit with. And I think, well, this is maybe also a part of the answer to how to share this type of data. Most probably it would make sense to get into contact with people who are already doing that. So, well, the Neuroglancer would come to my mind. I think CatMate also has an open access database. There's the Allen Brain Atlas. which I think can incorporate more data than they already have from various species. In Europe, there's the eBrains platform, which is mainly light optical data with much less resolution. However, there may also be ways to integrate EM data. But I think for the future, this is an extremely important question, because it's just too much data that one group can work on its own, at least from my opinion. So this is one thing that people are doing with it, imaging large individual data sets. Another one from our customers, Moritz Helmstetter at the MPI in Frankfurt, he went a different route. He's not imaging individual large data sets, but numerous smaller data sets. And last summer, there was a really nice science publication comparing different species, mouse, macaque, and human. And what they found was a quite significant change between mouse and primate in the interneuron to interneuron inhibitory networks. And so this is, I think, a nice way to also show the value of this type of research, because that's what people tend to ask all the time. What do you do with a large connectomics data set then in the end? What does it tell you? And I think a comparative approach between species, or also within the same species, but for between different individuals, or like mouse models for various diseases, I think this is what actually will tell you something in the end. And if it's about looking into the future, what can be done then? There are already plans for full mouse brain, which of course would be an even larger effort. Leaving alone the human brain, which is again very far out. However, sometimes things only need a starting point, and then they will gain their own momentum. So let's see. In the end, the solution, the technological solution, are in principle available. So the automated sample preparation techniques are there, and most probably more will come. We have the imaging technologies, we have the hardware for data storage and computation, the algorithms. So the processing is maybe the hardest part still. However, also this is research in progress, and I'm quite sure that in the next couple of years, more things will come out here. So at a certain point in time, maybe the full mouse brain is something that can be done. And at the moment, the multi-sem installations worldwide are, I would say, quite nicely distributed over the US, Europe, and Asia. So we have 3 in the US, 3 in Germany, and 3 in China, Japan, and Korea. And actually, 2 of our customers are already running 2 multi-sems, which of course is also a nice complement. If they already have one and they buy another one, then obviously they were happy with the first one. And I hope in the end this will help them to publish even larger data sets. And then maybe at some point in time, as I said, come to the whole mouse brain connectome. However, just trying to wrap the brain around these numbers is kind of mind-blowing, because in the end, it would be about an exabyte of data. So 1 million terabytes, 1 billion gigabytes, 1 trillion megabytes. So yeah, hard to imagine. Now, I want to close with, again, the takeaway message most probably is it's possible to image a square millimeter at 4 nanometer pixel size in less than 5 minutes. And this can be useful for not only neurobiology, but also reverse engineering of microchips or battery research or other types of materials research. And what I also would like to point out is there's a very nice overview of all types of volume EM techniques, the ZEISS volume EM, where you can also find a poster and a booklet in collaboration with Science. Either the QR code or the link will bring you there. And especially the booklet, I will quickly stop the presentation and show you. So this is the landing page, the volume EM page with all these different volume EM techniques, the poster which you can download, and this booklet. And the booklet contains a couple of short articles. I think one of the other authors is also in the call, as far as I know. And especially nice is that with each of these articles, there is a link to a short movie which recorded a workshop demo where you can get a better understanding of all these techniques in the video. And one of these articles is also dealing with a multi-sem. And in the video here, you can have a look at the full workflow where I just cut out a couple of snippets now for this presentation, but the full presentation would be in this video. And that's it from my side. And I'm open for questions.